Hi everyone, my name is Laure Wynants. I'm very thankful for the opportunity to speak here today. I'm going to talk about prediction research in a pandemic and discuss the results of our systematic review of diagnostic and prognostic models for COVID-19. From the beginning of this crisis, there has been a lot of interest in prediction models from academics, companies and governments. The UK government set out recommendations to 1.7 million vulnerable people to stay at home based on their predicted risk to get severe COVID. And in the United States, several algorithms to predict COVID-19 complications got FDA approval for use in clinics. So for clarity, when I say models, I mean any data-driven algorithm that has been developed to support decision-making for individual patients, for example, in making a diagnosis or prognosis, by combining several inputs and giving them appropriate weights, for example, combining symptoms and biomarkers. So for use in the clinic, such models could be translated into a decision tree, or complex algorithms can be incorporated in software, as is done here to automatically classify CT scans. Why are these models so important? Well, that's because we know from other diseases that a good model could improve care and reduce cost. So in this COVID-19 crisis, it would have been very valuable to help allocate scarce resources to those who would benefit most. The other side of the coin is that poor models can make matters worse. We know from decision analytic theory that models that yield inaccurate predictions can lead to harmful clinical decisions, worse than in the absence of a model. And risk scores that usually work very well to track a person's health during a stay in an intensive care unit did not work well during the 2009 swine flu pandemic, giving overestimated mortality risks. So we decided to start a systematic review. The purpose was to give an up-to-date overview and critical appraisal of all available models. We started this effort in March 2020, and in April 2020 we had our first BMJ publication with the results. Later on we published 3 updates and we're working on a 4th. We were able to work very quickly because we already had a toolbox available for the systematic review of diagnostic and prognostic models. The first tool in our toolbox was a CHARMS checklist. This is basically a checklist that gives you the key facts and figures that you need to extract from a paper when you're doing a review of prediction modeling studies. The second one is the Probust Risk of Bias tool. So this is a tool that standardizes critical appraisal by asking signaling questions in 4 domains: the participants, the predictors, the outcome you're trying to predict, and the analysis done to build and test the risk prediction model. In our search, we included peer-reviewed articles as well as preprints, but we only included preprints that hadn't gone peer review up until the second update. We searched for 3 types of models. The first types are the susceptibility models to detect those at high risk in the general population, among the people that are currently healthy. The second type are the models to diagnose COVID-19 or classify patients according to severity at presentation. And the third type are the prognostic models that predict the course and the outcome of the disease. So we did data extraction working in pairs of 2 researchers looking at the papers independently. And if more than one model was described in an article, the assessment of each model was done separately. So in terms of results, we've screened 37,422 records. From those, we included 170 studies. Together, they presented 236 models, of which 11 to identify subjects at risk in the general population, 118 diagnostic models, and 107 prognostic models. Most of these studies use single country data to develop a model, and the sample size was often limited to a few hundred patients. Most models were developed for use in hospitalized patients with confirmed or suspected COVID-19. And what's interesting is that almost half of them were available in a format that's ready to be used in clinical practice. About a third of the papers used logistic regression to develop a model. Another third used neural networks or deep learning. And then the other third used a whole array of different techniques. Top 5 most commonly used predictors in diagnostic models were vital signs, flu-like signs and symptoms, age, electrolytes, and imaging features. And in prognostic models, this was age, comorbidities, vital signs, imaging features, and sex. So what you really want to know, of course, is how well did these models perform? Well, within each type of model, we had models with an area under the ROC curve close to 1. This should be a cause for mass celebrations because it means that we can perfectly detect without a PCR test who has COVID-19 and who doesn't, and we can perfectly predict who will die and who will live. However, if you dig a little deeper, you will find that most models were not validated or only internally validated. Only a minority underwent external validation, meaning that they were tested in an independent data set of patients from another center or another country. We also found that only 4 models were low risk of bias, 6 were unclear risk of bias, and 226 had a high risk of bias. So this means that there were flaws in the design or the analysis that suggest that the estimated predictive performance is too optimistic. And you see that there were severe issues for all types of models and in all probust domains. For example, inappropriate study designs, ill-defined predictors, or using hospitalization for other respiratory illnesses as proxy outcomes. The analysis domain was most problematic, and this often had to do with small sample sizes. Combined with a data-driven modeling strategy, this often leads to overfitting and optimism in the performance estimates, and performance was also often assessed incompletely, looking only at the area under the ROC curve, but not at calibration, which assesses whether the predicted and the observed risks correspond to one another. But there was good news because there were 4 low risk of bias models, and these were the Q-COVID models. The Q-COVID models predict the risk of catching COVID-19 and then subsequently dying from it or being hospitalized with it. These models were developed on data from 910 GPs, and they had excellent discrimination and very good calibration in a validation in 80% of English adults. So have you been too critical? Unfortunately, validation studies confirm our fears that performance estimates were too optimistic. This plot is from one such study. The colored curves falling below the horizontal line shows that when tested, the models are less useful than oxygen saturation alone or age alone when predicting deterioration or mortality. Our research has had some impact. For example, the World Health Organization repeated our recommendation not to use any of the available prediction models for prognosis in the clinic. Like any study, ours also has a few limitations. One such limitation is that other models are available, for example, in-house developed models or proprietary algorithms that are not accompanied by scientific reports, so they fall outside the scope of the current review. However, there is some evidence that their quality is not better than the ones that we have reviewed. So in conclusion, despite reports of impressive predictive performance, much of the growing body of literature on prediction research for COVID-19 was of low quality. You should never trust a good reported performance alone. The study design and the analysis and the validation matter. This was a huge effort, and I have a huge team to thank. These are all the people that helped me in this work. If you would like to know more, please visit our website where details on each reviewed model are available, as well as a layman's summary written by COVID-19 survivors. I welcome any questions you might have, either during the Q&A or via email or Twitter. Thanks for your attention.